Thank you very much for coming to this seminar. We have the pleasure to welcome Félix Fracchia, who just finished his postdoc in our research group. Just back to the past to present Félix. He started his studies at University of Limoges in the field of biodiversity and the origin of populations. Then he did a master's degree in University of Dublin about genetics and evolutionary biology with a focus on human biology and evolution. Then he wanted to do a second master. So he went to AgroParisTech, but this time in Nancy. So he went back to France. And he focused on integrative biology of trees and cultivated plants. And in the same place at University of Lorraine, in collaboration with INRA and Nancy, he did a PhD degree in the role of phytohormone on the microbiota. Right. After this, he embarked on a postdoc with a co-founding project between Swiss Forest Lab and LabX, so between the French partner and VSL. And he accepted very bravely to work on this project, microorganism adaptation genomics, with Annegret Kohler from INRA Nancy, and Martina and myself here at WSL. So today we present his main findings and maybe some perspective about your future career. So thank you very much for coming, and I look forward to your talk.
Thank you very much.
Is it working? Yeah, okay. Sorry. So prevalence of water and nutrient availability in structuring fungal communities of healthy and decaying beech in forests. And the second part will be more dealing with the genomics adaptation of beech to water stress. So a little bit of introduction. So I guess we are all very much aware that we will experience an increase of drought event due to global warming, and it's what is represented here. So on the top chart here, you have the frequency of 2 consecutive years of extreme drought in Central Europe and under different scenarios. So depending on the quarter, so from the less bad scenarios to the worst scenarios. And what we can see is that we will have, this is very annoying, well, an increase of these frequencies of extreme drought. And we can see that the area of drought will also increase over time, no matter what the scenarios, and it's what you have here on the curves here. So if we now focus a bit more on beech forests in Europe, we can predict that we should observe a decrease of beech growth associated with climate change, and this under different scenarios. So it's what you have on the y-axis here. So less worse scenario here and a bit worse here. And this over time, so on the x-axis until 2090. And what we see is that we should observe a decrease of beech growth, which is what you have here on the red quarter in all Europe, except for some different little parts in the northern continent, where we should, on the contrary, observe an increase of beech growth. Yet, we also know that forest trees are a huge ecosystem and they are interacting with a cortege of microorganisms. And this is the so-called microbiota, right? And in this microbiota, we can find different kingdoms. We have fungi and oomycetes, bacteria, viruses, archaea, and protozoa. And in this talk, I will mainly focus on fungal communities. This microbiota is very important because it's involved in the extended phenotype of the tree. And indeed, they play an important role in biogeochemical cycling and through the decomposition of organic matter, and it's leading to a role in the development of the host. And indeed, through oligonutrition, for example, or bioavailability of different minerals. And what's going on here is that this microbiota is able to transfer phosphorus or nitrogen, for example, so nutrients to the host in exchange of phospho-assimilated carbon from the host to this microbiota. And this is very important because this transfer can reach up to 30% of the photo-assimilated carbon of the host to its roots and to its microbiota. So this is one of the first roles. Another role of the microbiota is that it can protect the plants against different stress, such as biotic stress with pathogens attacked or insects attack, but also against abiotic stress, such as drought or extreme temperature. And it's what we will focus more on this talk here. So if we now have a look at the different mechanisms that can regulate the assembly of the microbiota, as I said, we know we have abiotic factors, such as the climate, seasons, but also soil properties. If we talk about soil texture, availability of nutrients, pH, presence of pollutants. And we also have biotic factors that is regulating the assembly of this microbiota with an increase or gradient from the soil to the endosphere of the host, where the host is playing a very important role here. So we know that from the soil to the rhizosphere, there is a first selection step due to root exudates. So it will attract or repel different microorganisms. And then we have a second step from the rhizosphere to the endosphere, where here the influence of the host is the most important. We also know from different studies that these fungal communities will also respond to global warming. So here I just presented the main factors that will influence these communities. So we know that we have warming, drought, nitrogen deposition, and elevated CO2. And if we focus here, for example, on warming, we should observe a rise of pathogens, but also reduction of ectomycorrhizal diversity. And if we now focus, on nitrogen deposition, we should also observe a decrease of the ectomycorrhizal species that are using organic nitrogen, because we will have more nitrogen available, so less use of this ectomycorrhizal species. Yet in this study, they didn't take into consideration the plant host. And as I just showed you before, the host is also playing a huge role in the establishment of these communities. And as the plant host will also be affected by this global warming factor, of course, it will then indirectly influence the microbial communities. So here, it's where we come from with these two main research questions. They are very much related one to another. So the first one is that we were wondering what is the main driver of the assembly, environmental driver of the assembly of fungal communities associated with beech roots in forest. So here we look at different environmental parameters such as the climate, nutrients availability, pH, etc. And in parallel with this, we also wondered if we have specific communities that are associated with healthy or decaying beech across a water stress gradient. So to answer these questions, we used a very nice sampling design over Switzerland, where we have 6 sites, so distributed across Switzerland. And for each of these sites, we have the measurements of more than 150 environmental parameters over 10 years and at different soil depth. When I'm talking about variables, we can put them into different categories. So we have climatic parameters such as the air temperature, soil temperature, vapor pressure deficit. We also have vegetation and terrain attributes, such as the leaf area index and topographic attributes, such as the slope, valley depth, etc. And finally, we have also edaphic parameters, so soil properties such as mineral content, pH, cation exchange capacity, a lot of them. And of course, soil water potential, and I will go back to this in the next slide. But what we did is that we sampled pairs, 10 pairs for each site of healthy and decaying beech trees. And what we did is that we sequenced the fungal communities that were associated with the root system of each pair. So this is just an example of what the trees looks like when they were sampled. And here I just would like to go back a little bit on soil water potential, because these are very interesting studies that had been done by Laurent Walzert. And what he did is that he related the soil water potential to leaf water potential to be able to put some different thresholds that were related to different stress that was experienced by the tree. So to be short here, from 0 to minus 0.2 megapascal of soil water potential, there is no stress. But when we cross this threshold, we observe moderate stress from the tree. So from minus 0.2 to minus 0.8 megapascal, we have xylem, embolism, and defoliation. Then we can go to high stress. Oh, sorry, this is for high stress. For moderate stress is reduced soil water uptake, stems free concentration of growth. High stress, we have xylem, embolism, and defoliation. Very high stress, we have a reduction, 90% reduction of stomatal, maximal stomatal conductance, and nearly zero transpiration. And finally, we can see very high stress when we are above minus 2.1 megapascal in the soil. So knowing this, we had the record, as I said, over many years of soil water potential in our 6 different sites. So we use this, and it's what you have here. So for Tamins, Neukrich, Buren, Hemmingen, so these are the 6 sites. You have soil water potential here on the y-axis and years on the x-axis. And what we did from this is that we derived an index that we called integrated drought index. So it's the integral of soil water potential depending on the stress threshold, right? So if I highlight this, maybe it will be easier for you to understand, is that we took this stress, so here it's not stress, sorry, yeah, and here trees are stressed. And we use this as a integrated drought index because it's a composite index that goes from the severity of the stress, of plant stress, beech stress, sorry, but also its duration and frequencies, right? And according to this, we were able to show that we have a gradient of stress in our 6 sites going from the less, so the wettest one, if you want, which is Tamins here, and to the driest one, Chamozon in red here. And I will keep this color code for the rest of the presentation. So bluish color will be associated with the wetter sites and reddish color to the drier sites. So if we jump now to the results, out of the 150 variables that we used, only 4 were mainly explaining the distribution of the fungal communities. So it's what you have here on this RDA. So for those who are not familiar with this kind of representation, each point here represent the fungal communities associated to a sample. You have the color code, as I said before, so from the wettest one in blue to the driest one in red, and the health status of the host. So healthy are represented in square, and decaying trees are represented by circles. What we can see is that the distribution of the fungal communities is mainly driven by sites, right? It's driven by the colors here. It's not driven by the health of the tree. This was very surprising for me, but yet. And out of these 150 variables, as I said, we had 2 drought index, integrated drought index, so 2 water stress related parameters that explain most of the distribution here. And when we extract the variance using variance partition analysis, this is the results we get. So basically, as I just mentioned, environments, so the different environmental parameters we use explain 90% of the explained variance. And out of them, ID, and the ID in the deep soil explained more than 12%. So most of the distribution, most of the explained variance is due to these drought-related parameters. And then we can move on to the richness of the arboreal layer, organic phosphorus, as well as age, and finally health of the beech that accounted for less than 1% of the explained variance. We can go a little bit deeper now and trying to see if we can correlate yields with these environmental parameters. So you have the different fungal yields here. So ectomycorrhizal in blue, endophytes in yellow, pathogens in green, and saprotrophs in brown. What we can see is that we tend to have a correlation between healthy trees and ectomycorrhizal communities. And on the opposite, we have saprotrophs and pathogens that are correlated with decaying trees. Well, so far so good, nothing very new. But another thing is that ectomycorrhizal species are negatively correlated with the amount of organic phosphorus. And this is for me super interesting. Nothing new again, because we know that ectomycorrhizal species are providing phosphorus to the tree. So when phosphorus is abundant, there is no need of ectomycorrhizal species, right? So they are negatively correlated to it. Okay, so this is for the beta diversity. If now we move a little bit on the composition, what we see here, so you have the different relative abundance of the most abundant species, sorry, and we can see that these three groups, basically. So one first group that is here in orange, that is always present, no matter of the stress gradient. So sorry, I should say that on the y-axis, you have the sites, so from wettest site to the driest site here. And each square here, you have the healthy and decaying trees for all of them, right? So we have, just visually, we have species that are always present, no matter of the stress and no matter of the health. We have another group here that tends to increase with the increased water stress gradient. And on the contrary, we have species here that tend to decrease with the water stress gradient. And it's what we want to dig into now. And here, if we look at the richness of these communities, what we see is that there is no influence of, so for alpha diversity, there is no influence of water stress on the richness and on the evenness of the species. But what we observed is that we have a significant increase of OTU's richness, sorry, from healthy to decaying trees. So here is the color code again. Sorry, maybe I should have mentioned this first, that the light color here is corresponding to healthy trees, and the darker color is associated with decaying trees. So they go by pairs again, all the time. And what we see is that we have an increase of OTU's richness from healthy to decaying trees. And this for all the sites we study. If we now move to the different yields, the roots were dominated by ectomycorrhizal species that accounted for more than 34%, followed by saprotrophic fungi, around 26%. And we, again, had no significant effect of water stress on these yields, right? But what we saw is that we have a decrease. So it's the same color code again, light color for healthy communities, well, communities associated with healthy trees, and darker color associated with decaying trees. And what we saw is that we have a significant decrease of ECM from healthy to decaying trees, and this for all the study sites. And on the contrary, we had an increase of saprotrophic fungi from healthy to decaying trees. So this goes back to the RDA I showed you before, but it's a bit more precise here. And we had no effect that we could observe on pathogens and endophyte species. So finally, what we wanted to do is to identify species that were significantly associated with this water stress gradient. So we used linear discriminant analysis effect size, LFSE. And we were able to identify 18 fungal species that were significantly associated with these different sites. And it's what you have here. And what is interesting for me here is that, again, we have this no link with guilds, depending on this. It's only depending on specific fungal species. And what is striking for me and in supports our finding is that these guys that were associated with the driest sites were positively correlated with our drought index also. Whereas the guys that were associated, fungal species that were associated with the wetter sites were negatively correlated with this drought stress index. And here, just to give you the name of two species that I think are very interesting, you have Oidiodendron here, you have two of them. So they are endophytic species, and they are very much associated with the driest sites. So I think they are of much interest. And you have also Lactarius blennius here. And this guy is an ectomycorrhizal species, and it's a specific ectomycorrhizal from Beech. And I think this needs further investigation, but the fact that he's here significantly associated with the driest sites are, I think, very of interest. We did the same thing, but here, instead of looking at the water stress gradient, we looked at the vitality of the host. And we did not find any species that were significantly associated with healthy trees, but we found 4 saprotrophs that were associated with the decaying trees. And it's the 4 guys you have here. And again, we have 2 Pesicula radicicola that are significantly associated with decaying trees. Okay, so this was for the first part. And I will now move on to the genomic signature of adaptation in European beech population, of course, water stress gradient. And I think this picture illustrates quite well the problematic we have here, except that this is for oak species and not beech. But if you look at these species, these trees, they are all growing in the same environment, but some of them are healthy and some of them are decaying. And so the only explanation we can have is that it's come from the genotype of the tree here, right? So little bit of introduction again here. If we look at phenotypic plasticity and adaptive process in response to climate change, here is a conceptual illustration of natural selection in the context of anthropogenic climate change. So if we look at, so you have the fitness here on the y-axis of the and each dot here represents the populations. And what we see is that if you look at environmental condition, we have an optimal fitness that has been driven by stabilizing selection, right? If we now move to the current environmental condition, we know that climate change induces a shift in the fitness. So we have a decrease fitness here. And if we now predict it or project it, to future environmental condition, we know that natural population will be severely depressed in fitness and have a risk of extinction because their fitness starts to be very, very low. So how to measure local adaptation? So these 2 main ways to do it. It's common gardens and reciprocal transplant experiment. It's difficult. And just to illustrate this here, again, you have the fitness on the y-axis, sorry. And here you have 2 populations. One is coming from south and one is coming from north. So if you put, and we assume that each locally adapted population in its native site has higher fitness than any other population in the same site, right? So if you put the south population in south condition, they will have a higher fitness than the northern population. And on the opposite, if you put northern population and, well, if you put these 2 populations in northern condition, northern population should have a higher fitness. Another way to think about it is that you have different fitness due to different alleles, right? And so you have Cs here, as for example, that are beneficial allele for cold condition, and Ts that are beneficial allele for warm condition. And again, if you put this population, you should have the Cs that have higher fitness in cold condition and Ts that are more higher fitness, in warm condition. And this kind of thing can be very unrealistic for organisms with long-lived generation time, such as forest trees. And so another way of doing it is to use genotype environment associations, so GEA, which assume local adaptation of population. And here we look at the population allele frequencies of Cs here under a temperature gradient, for example, on the x-axis. And what we see is that if we are in cold conditions, the beneficial C allele should be more frequent, right, than if we go to warmer conditions where Ts should be more frequent, but we measure Cs here. And in the middle, you should have different SNPs, right? And so this is this kind of analysis that is called GA. And so going from environment or genotype to environment, and it associates genetic variation with environmental condition. And it allows to identify allele frequencies that are associated with environmental factor, and thus identify environmental factors that are underlying local adaptation. So in order to do this, you need a very nice sampling design. You need to have environmental data, of course, and you need to have your genetic data. So there's a lot of bioinformatic and field work here. And then you can do your environmental association analysis using regressions, mixed model, well, there's different ways of doing it, and then post-hoc validation. So here we had two questions. The first one is Beech exhibiting genomic signatures of local adaptation across a water stress gradient? And the second question is that are fungal communities derived environmental predictors associated to genomic variation in beech population? So to answer this question, we use exactly the same experimental design as I presented before. So it's exactly the same sites, exactly the same environmental parameters that we measured. And instead here of sequencing the fungal communities, we sequence the whole genome of the host. So it's exactly the same trees, right? But except of having the fungal communities here, we have the genome of the host that is holding these fungal communities I presented. And here we did the SNP calling, and we ended up with more than 240,000 SNPs. And the beauty of this work here and the great idea of my supervisors is that instead of just using environmental parameters, as I presented before, we also used the fungal communities that I presented before, right? So we used like the relative abundance of ECM, relative abundance of the different alpha diversity index, or everything like this, and we use this as biotic factors, biotic predictors in our model. Okay, so first thing we need to do when we do this kind of analysis is that we have to check for the structure of the population, because natural population structure can mimic genomic signatures of adaptation. If you have a bottleneck, for example, it will lead to a kind of genomic signatures that you will think it's due to local adaptation, but it's definitely not due to this. It's only a bottleneck. So here you have so we did the PCA. So here each dots represent not the fungal communities, but it's the SNPs, right, for each sample. You still have the color code as I explained before. And what we can see is that we have a very shallow structure because on all the samples, are grouped in the middle here, except for two sites, Tamins, so the wettest one, and Chamozon, the driest one here, if you look at the two first pieces here. And if you now look at the third pieces, with the first PC, we still have this split between Tamins, Chamozon, Neukrich, and we have this Remmingen that is going out a little bit. But let's say the structure is not that clear here. There is another approach to look at the structure, and it's called sparse non-negative matrix factorization. So it's SNMF analysis, and it investigates the ancestry coefficient. And it's what you have here. So we have K equal 4, sorry. And what we see again is that we have this chamozon, so the driest one here, and tamines and licorice that seems to differ a little bit from the others. And we have this group that tend to be very similar. And we use the third approach that is called admixture, still with K equal 4. What is nice here is that you can plot it on the map. But we obtained exactly similar results from SNMF and the PCAs that I showed before, where we have tamines and chamozons that tend to differ. So it's again this one and this one. And we have a group here that is most similar, except that here we have licorice, we have 2 licorice sites, and so they are overlapping, so we cannot really see. But this one is different from the other one. Okay, so now that we have assessed the, it's very dirty, sorry. But now that we assess the population structure, we can now move on to the GEA analysis. And I will just try to be short here in the methods. But basically, we used the different environmental categories that I presented before. So we have abiotic one, such as soil chemistry, topography, climatic, and biotic categories, vegetation, fungal composition, yield, and diversity. And in each of these categories, we have different environmental predictors, right? So for soil chemistry, basically, we have pH, CN ratio, cation exchange capacity, soil depth, etc., etc. So each of these categories is one model, right? So we basically have 7 models. We run 7 different models here, one for each category. And we used multivariate LFMM, so multivariate latent factor mix model that is using linear regression. And we added on top of this another linear regression to be able to decipher the influence of each variables here. So what we looked here after running the analysis is the number of significant association, the strength of this association, so the effect size of the beta, and as well as the importance of the model, which is the adjusted R square. So if we now look at the number of significant association and the effect size based on categories, what we see is that you have the number of associations that has the gray square here and the effect size, so the strength of association in the white square here. You have the different categories on the x-axis and number of association on the left and strength of association of the right. And what is striking is that we can see that fungal composition, yielded the most important number of significant association. We have more than 1,200 significant association when we used the fungal composition categories, right? But it has the weakest strength of association. Too bad. On the contrary, if we look at topographic category, we can see that we have a bit less than 200 significant associations, but it exhibited the highest strength of associations. So they are very, very good associations, let's say, even if we have less the less number of significant associations, sorry. So once we have here, as I said, here we are looking at categories, but we can go a little bit deeper into the model and look if we have one variable in these categories that is driving all what we can observe here. But that's not the case. So basically, if you look at fungal compositions that are here on the left, we can see that all of them, so Oidiodendron, as I said before, or Lactarius blennius, so the 2 fungi, if you remember that I shortly described before, they are the ones that exhibited more than 500 significant association and still with a very low strength. And if we go to topographic now, so if we look at slope, which is one, yeah, it's the one that exhibited the highest strength, but it's also true for the different topographic index, such as northern here, and we have the curve profile here. Another thing that is interesting is that diversity index, such as Shannon and richness, so alpha diversity for fungal communities, we had very, very low number of significant associations, but they were quite high in strength also. Okay, So once we did this, we can also plot the significant associations depending on each categories. And it's what you have here. So this is called the Manhattan plot. Each dot here represents a SNP, right? So you have 240,000 SNPs. And when it's above this dotted black line here, it means that it's significant. So there are the significant associations, right? So as I said, for fungal composition, and sorry, you have the different chromosomes on x-axis here. So 12 for Beech, right? From 1 to 12. So these are all the significant associations for fungal composition. And if we look at guilds, now fungal guilds, we can see that we have 1/4 of this association because we only have around 300. And what is interesting here is that we can also annotate these genes and to know what they are encoding for or which region all the SNPs are. So sorry, it's a bit messy here, but it's just to show you that we have the data because it's whole genome sequencing to be able to know where are the SNPs and what are they encoding for. And I will go back to this in a few slides. It's just to show you that here you only have the top candidates. So I think I put the top 10 genes annotation from the top associations that we observed. So this is for, again, fungal composition and fungal guilds, but we can also do the same with abiotic parameters, such as deep soil and topsoil climatic parameters. You can see that we have much less number of significant associations here, and say similar thing for vegetation history of each site. But once we do this, another thing we can do is that we can see what these SNPs are doing in the genes, right? Is it a synonymous mutation? Is it non-synonymous mutation? Is it going to lead to modification of the protein? Is it going to make the protein not functional anymore? And it's what we did here. Oh, I'm sorry again. So basically, most of the high, so here I highlighted all the high impact on the proteins, because if the modification has no impact on the protein, well, it's still functional and nothing changed in the function, right? So here I'm only focusing on the SNPs that were linked to high modification of the proteins. And what we can see is that most of these modifications were due to a stop codon in the encoding sequence. And what I think was super interesting here is that we have these nin-rin-like proteins. So I have absolutely no idea what it is doing, but it's coming out a lot in these fungal communities categories. But what I think was even more interesting is that if you look at fungal composition, we have a lot of significant associations that are involved in sucrose synthase and in disease resistance protein. And from what I said before, I think this is super interesting that we can highlight that a lot of these modifications are involved in sucrose synthase, because if you remember, we have a transfer from the plant to the roots of photoassimilated carbon, right? Under the form of sugars, yeah. Okay. So this was for fungal composition, and if we now move to abiotic factors, here I'm only putting climatic one in the soil, sorry, and we observe a lot of retrovirus-related pole protein and transposon, but for both actually, sorry, and in these abiotic factors, they are much more related to the integral component of the membrane of the plant. Okay. Another thing we can do is called genome-wide association analysis, GWAS. And this is to identify loci that are significantly associated with phenotype variation in our beech populations. So instead here at looking at the environment, we will look, I have time? Yeah, good. We will look at the phenotype of the tree and its fitness, right? And here, when we talk about phenotypic traits, we use two phenotypic traits, one that is binary, so from healthy to decaying, just categorical variables. But we also have a continuous variable because we have measurement of data 13C in each of these trees. And what's going on with delta 13C is that we know that when the tree is stressed or decaying, we have an increase of delta 13C because it's closing its stomata, right? And so we have an increase of delta 13C, what we don't observe in the healthy trees, right? So we test these association between genetic variants and phenotype variation, sorry. And we use different models. So I will not go into the details here. I will just jump to the main results. Is that so it's again the same thing as before. It's a Manhattan plot. So each dot here represents the SNPs, and we have two different thresholds. And when they are above the threshold, it means that they are significant, right? So for the health status, so the categorical variable, categorical trait, healthy, decaying, we did not find any significant association. But when we looked at delta 13C, so the continuous variable, we observed a very nice peak here on chromosome 2 that is highlighted significant associations here. Sorry, here. But we did not annotate them yet. So with this, I will try to sum up quickly. So for the first part, we showed that water stress-related predictors, rather than the other environmental parameters we looked at, were the main parameters that is structuring fungal communities. We showed that even if the health were only accounting for 1%, well, less than 1% of the structure of the population, which I think is very striking. But we could see that the health status, but not water stress, was influencing or increasing, decreasing the richness of fungal communities. And ectomycorrhizal and saprotrophic fungi were also associated with health, positively correlated with health, but not with water stress. And we could identify specific fungi that were associated with the dry sites as well as decaying trees. If we move on now to the genomic part, we showed that we had both biotic and abiotic predictors that had significant associations, in the GA analysis. We showed that the fungal composition category exhibited the highest number of associations, but with the weakest strengths. And conversely, we had the topographic category that exhibited the highest strengths, the highest beta, especially the slope, which is a proxy of drainage of the water, right? Most of the significant association were related to component of the membrane or ATP binding. So this is from gene ontology analysis. And as I said, most of the high impact on protein were due to stop codon. And I would like to highlight again this finding is that we had sucrosyntase and disease-resistant proteins, that were mostly associated with fungal communities and guild categories. And finally, for the GWAS, we had a very nice peak on chromosome 2. And so this, we need to annotate these SNPs and to see if we can find similarity between these guys and with SNPs that we detected in the GA analysis. Okay, so I would like to thank you very much. I would like to particularly thank my supervisor. So Anne Koller from my group at INRA, and Benjamin and Martina from the group at WSL, and of course Sabine and René, who did a lot of sampling on the field and DNA extraction and everything. Laurence Walzert, he's a guy who provided all this amazing data on the environmental categories. André Baltensfehler, for all the topographic, I'm not used to these names there, sorry. For all the topographic data, and of course, Félix Gugerli, Christian Helstab, and Olivier François, who is a very, very nice statistician and who developed all the tools for GA that we used. And I'm happy to take any question if you have some.
Thank you very much, Felix, for this great talk. Is there any question in the audience?
Hello, thank you so much for your talk. It was very interesting. I'm curious about, you categorize your beech as healthy and decaying. What do you call decaying? Is it like dying, or is it like signs of decay in the wood, or is it dead fully, or is it?
So they were, sorry.
Sorry, go
No.
ahead.
So no, they were still alive, and they were categorized as decaying when they lost more than 80% of their leaves, if I remember correctly.
So it's a dying stress tree.
Yeah,
It's not.
yeah. They are on the way to die, but they are not dead yet. But when we did sections, well, we accounted for the age of each of the trees. I didn't talk about this, but some of the dead ones, you could see that they were little, I don't know, rotten parts. So they were starting to really decay in the core wood. Yeah, but not all of them, right? On the section, you could see that parts were still alive, so they were still having some flow of xylem and phloem and everything.
Thank you.
This is working, yes. Thank you very much. So I have a related question next to this one. So do you think that the differences in terms of percentage of composition of abundance of the ECM, for example, between healthy and decaying trees are a cause or a consequence of the healthiness of the trees?
So this is the egg or the chicken question, right? Yeah. So from this, we cannot answer. Because these studies cannot answer this question. We don't know. What should have been done is to sample trees before they were decaying and after, but you don't know if they will decay or not. So it's very difficult. My opinion is that it's not a cause of the decaying of trees, because if you look at the saprotrophs that were associated with decaying trees, the relative amount, well, first, they are not pathogens. Saprotrophs are able to degrade carbons, right? But we only had 4 species, and the relative abundance is not huge. So then maybe people working in phytopathology will have different answer than this. But for me, when you have a tree that is decaying because of fungi, the relative abundance of the pathogen or the saprotroph is much higher than what we could detect, right? So I would say that the tree are decaying, and thus saprotrophs are coming more, and ectomycorrhizal are going out, but are replaced maybe by saprotroph. But I would not say that they are decaying because of saprotrophic fungi. I would say it's rather consequence than a cause, if it's answering your question, right? Yeah. But just one last thing, we had Hymenopilus fungi associated as a saprotroph, significantly associated with decaying trees, and this fungi is able to degrade carbon in a live beech. So, but we cannot really say for sure that it is. Yeah.
Yeah, maybe a question from somewhat out of the field, but still related. I mean, you made the conclusion that soil properties do not matter that much in your study, and of course, that rings an alarm in my field. I mean, you only looked at calcareous soils, I've seen, so they are per definition probably similar. Or I mean, you also have probably chosen the sites to be similar in soils.
So that's a very good question. So I didn't choose the soil. I had all my confidence in the other people for doing so. So say again your question.
No, I mean, just from recognizing the sites, I mean, they all are calcareous, or I mean, they don't span a gradient in soil properties, so they are probably all similar. Or I mean, at least you should explore this.
But
show it.
Yeah. So I know that they were selected. I know the pH is rather similar. Then for clay content, I don't remember exactly, but I would say we have a gradient. But these sites were specifically chosen because they were really dry, in fact. I showed the gradient, but they are on the edge of the gradient while they are very, very extremely stressed. And it's also why we chose them is that we wanted to have decaying trees and healthy trees. If you go to a site that
yeah, it's totally okay. But I think I also would do it like you. Huh? I mean, I would go on one soil type, which is the same, but I would not draw the conclusion that actually soil does not matter in this context.
I'm not saying it doesn't matter. No. I'm saying it's not the most important driver. But of course, if everything is homogeneous, you will never see that there is an influence of the soil. This I totally agree, yeah.
And also I would be carefully with this. Organic phosphorus is not the indicator for nutrient availability.
Okay.
Just
No, it's good. You can criticize as much as you want. I'm here for this, so don't worry. Thank you.
Hi, thank you for the nice talk. I also have a question about the first part of your talk. The sites that you selected, each one of them show a slightly different drought index, no?
Yeah.
And then you are showing us this very nice ordination analysis where the sites are basically separating the fungal community. And you are concluding that most of this effect is because of the drought, no? But if each of the sites has a different drought level, how do you know it's the drought and not something else? Like fungal community tend to be very local, no? So could it be just a site effect? Or?
It's a very good remark, but something I forgot to mention is that to do this, we only assessed the fungal communities that were present in all sites. Yeah. So if we only took into account species that we could detect in all the sites. Yeah. So it's only the relative abundance of the species, but not the presence absence of the species. Yeah.
Okay, thank you.
So maybe related to this, you mentioned that you have an increase in the OTU richness.
Yeah.
If it's relative abundance, how do you?
Well, OTU's richness, it's only presence absence of the UTU.
Yeah, but if all the species are present in all sites?
No, not for the diversity. Diversity is everything. Okay. For the diversity, we did not select only the species that were present in all sites, because of course the richness will be exactly the same. Yeah. So for not for this, not for alpha diversity, we did not select anything. We took all the communities that were present. Make sense or not?
Yeah, but then so what was
the question was related to the RDA, ordination plot. Yeah. But yours is related to alpha diversity.
Yes. Okay. Sorry.
No, maybe my answer is not. Yeah. We can talk after if you want, it's no problem.
Yeah, I also have a question to the ordination plot. So do you think that the fungal communities are directly driven by the drought, or do you think it's like an indirect effect of root exudates, or like, you know, the reaction of the trees to the drought?
It's multifactorial. Yeah. Of course, if your trees are stressed, the root exudation will be different. Maybe it will allocate less carbon to the root. So there's many things. But this we did not measure, so we don't know. The only thing we know is that from environmental parameter, the ones that explain most of the variance, so most of the distribution of the communities are two drought-related parameters. We're not saying that soil is not important again, no?
There any other questions, maybe in the chat? No? Okay, so there is a little bonus over there.
Similar related to the between sites, did you look into variation within each site for both biotic and abiotic parameters?
I don't understand the question, sorry.
Do you have data on variation within each site of like biotic and abiotic parameters you measure?
No, for each site, we have only one value. We don't have the pH for each sample, for example. If we have 10 trees in one site, we only have one pH value.
Okay.
Yeah. Is it your question? Yeah.
Yeah.
No, it's pseudo replicates for this. it would have been much better, but yeah.
All right.
Yeah.
If there is no further questions.
It's only for environmental parameters
Yes,
where we only
so
have
there
one.
is a discussion. You want to discuss it? Before we close, if you want to join the table that we have reserved for the discussion, then you are welcome to join. But I want to thank you again for coming to WSL. It was still a 5-hour drive to come here.
No, it was less.
It was less, so thanks so much. And again, congratulations for the completeness of your postdoc.
Thank you.
Thank you. Well done.